How many completely different amino acid sequences might give a similar three-dimensional structure for 150 ways to long proteins? Answer will be 10 to the 26 different sequences might fold into single similar structure. The basis for this conclusion is something like this. The total possible sequences will be calculated by this equation, assuming there are only 20 different amino acid type. That will give you 10 to the 200th magnitude, and the totally different non-homologous proteins, assuming that the protein sequences with less than 20%, the similarity will be 10 to the 38th magnitude. Generally, it is said that 1 out of 10 to the 9th magnitude is a stable structure, so that the stable proteins might reduce to 10 to the 29th magnitude. How many different topologies are possible? Currently, probably around 1000 types, so that for each topology, different structures, how many different sequences? 10 to the 26 magnitude sequences. How to predict the three-dimensional structure of a protein from its amino acid sequence is a major unsolved problem. It could be approaches by experiment or computational simulation by using computer. There are so many different approaches in this field, but two things are introduced in this textbook. First, the inverse folding. This one is based on which sequence patterns are compatible with a specific fold. But still, there are too many possible sequences for single fold, as we learned in previous slide. So it seems like this is not practical. So another approach is called threading. So in this issue, just addressing which of the known folds, if any, is most compatible with a given specific sequence. So this is more practical. First, calculating the possibility of a given sequence for each known structure, and then select the highest possible structure. Protein engineering can be used to increase the stability of industrially important enzymes. There are three approaches were made. We're going to explain first the mutation which decrease entropy of unfolded state. this is just a matter of less number of possible conformation of unfolded state. In this picture, the T4 lysozyme engineered to introduce new 3 disulfide bond as shown in yellow color here and here. So disulfide bonds just stabilize by decreasing entropy of unfolded state. Likewise, mutation such as the replacement of glycine with any other residue or any residues with proline also could increase the protein stability for same reason. Another important aspect of this kind of experiment, whether the contribution of mutations at different sites to the protein stability could be either additive or synergistic. Additive effect means each different site contribute independently to the stability of this protein. In the case when they combine, such mutation, multiple mutations are combined into together into a single protein, the effect will be additive. Melting temperatures Tm of engineered single disulfide bond containing mutant of T4 lysozyme relative to wild-type lysozyme are shown here, left 3 bars, where reduced form of lysozyme versus oxidized form, meaning containing disulfide bond. So you can see the increased stability by introducing disulfide bond for 3 different mutants. In right side you can see doubly or triply mutated proteins. The red one is obtained by measuring experimentally, and the green bar was obtained by just simply mathematical calculation of each single mutations based on single mutant values. Next strategy is to use stabilized dipole of alpha helix. In chapter 2, we learned that alpha helix can be considered as dipole with a positive charge at each N-terminus and a negative charge at the C-terminus. So introduction of minus charge in lysozyme at two positions, here and here, both at the end terminal of two alpha helices, by substituting asparagine or serine with aspartic acid, lead to increase of stability in this protein. You also saw potassium channel, the position of C terminus of alpha helix near to potassium channel could attract the positively charged potassium. Third principle is to increase hydrophobic interactions, especially in the interior core. So you see the cavity found, which is very rare normally in protein internal structure, could be filled with the hydrophobic side chains by replacing residues with the residues with larger hydrophobic side chains. So filling the empty space cavity with hydrophobic side chains end up with the increased stability. But you must remember two things regarding the protein stability engineering. Knowledge of the three-dimensional structure of the protein is a prerequisite to engineer protein stability. Secondly, as yet, only 50% success rates are reported, even with very careful selection of mutation sites, because protein structures are very delicate, so that the mutation could result in some delicate structural disturbance, even the position which is very far from the mutational site.